compute. Good. Okay. Thank you, Catherine. First of all, thank you, Catherine, for taking time for this. I assume you're very busy, probably busiest that you've been in your life. So thank you for doing this. Thank you for your work, of course. Everybody else, thank you for joining. My preamble will be very short. Why? Because I have 7 pages of questions for Catherine. Some are mine, some are yours. And we want Catherine to go to sleep before midnight. So briefly, Catherine is associate professor in the School of Medicine at Stanford University, and her specialty is infection disease. Now, if you registered, you were on a page where it had the long bio of Professor Blish. And myself, I will skip the introduction. If you have been at a laser, you know who I am. If not, well, the lasers are on hold, the physical ones, and we replace them with these dialogues that I call the last dialogues, life, art, science, technology. Hopefully, of course, they are not the last ones. Oh, vanity. I originally invited Professor Blish in mid or early February. And I think by then, 0 people had died in the United States. And of course, we had no idea that today we would be talking after 110,000 deaths in the United States. I don't know, many worldwide, 400,000, whatever. Originally, I was just interested in viruses. So I wanted to understand better the life of viruses. Now, Professor Blish will correct me. And so let's get into it right away. So first of all, so I'm in a room. How many viruses are here? When I'm in a supermarket, how many viruses? When I walk, I hike in the forest, how many viruses? How many of these things are we surrounded? And how many species? And how many of them, how many of these species are bad for us?
So viruses are everywhere and all around you. How many viruses there are is an interesting question, probably in the trillions, if not trillions. But the vast majority of those don't infect people, they infect bacteria. And in particular, and in the ocean, there's like this incredibly rich life of viruses that are just in seawater, which don't freak out next time you go swimming. But it's, I mean, they're all over the place. But of all those trillions upon trillions of viruses, there are probably only about 250 or so that can actually infect people. So each virus is very specific to its host.
Are some viruses good?
They know us better than we know ourselves, so.
Sorry, are some viruses good for us?
It's a good question, probably. Just as I think hopefully a lot of people in the audience have heard of the concept of the microbiome, all the beneficial bacteria that live within us. And, there's tons of bacteria in our guts, on our skin, in our mouth, every place. And we live in a symbiotic relationship with that, and it's beneficial to our health. And there can be better or worse bacterial flora. But an area that's really only just beginning to be appreciated is that those bacteria come with a lot of viruses, and the viruses that infect the bacteria. And this is a completely understudied area, but a few people here and elsewhere in the world are looking into it. And one really interesting question is, are there viruses that are beneficial because they're maybe holding a certain bacterial species in check, like a bad one, or promoting the growth of a good one? And really, we don't know much about that whole diverse world of what is termed the virome as opposed to the microbiome.
The virome. Okay, so now, how do viruses evolve? How often they mutate? And can you clarify this thing? I mean, viruses, we read about the genome of the viruses, but you scientists telling us they're not life, but they have genes. Can you clarify this whole picture of how they evolve, how they mutate, and that at the end of the day, they have genes?
Yeah, well, so they have genes because they're pretty much mostly genes. So I don't know if you read, or if many of the people listening read, the New York Times article I think it was over the weekend, on where we're at with COVID, and they described it as a photocopy machine. And I thought that was a really well done article that people should find if they haven't, and it actually covered a few of the questions that we've already discussed. But so a virus is not considered alive because to be alive, you have to have metabolism, and you have to have the ability to be able to reproduce yourself as a species. And viruses themselves can't do any of that. They're basically a sac with some genomic material that carries with it the ability to know how to make more of the sac and the genomic material. So, and that genomic material can be either RNA or DNA. And the sac is typically a complex of proteins that basically are the carrying card that tell it what cell it can enter. And so you'll have little proteins sticking out on the surface, and that will bind to something on a cell and allow it to enter through all kinds of different beautiful complex mechanisms. And then once in the cell, the virus basically hijacks that cell to make more of itself, and the process repeats. But without a cell, a virus is nothing in terms of, it's just some proteins and some DNA or RNA. And some envelope.
So first of all, to get an idea of the magnitude, how many genes are we talking about for a virus?
Oh, it's incredibly variable. I mean, the smallest viruses are the parvoviruses, which only consists of about 5,000 base pairs of DNA, which is really tiny. HIV, where I did a lot of my work, all retroviruses have 3 major genes. One is called the polymerase. That's how it makes more copies of the genome. The GAG is the capsid protein that actually kind of self-assembles around it. And the envelope are the little spiky things that allow it to enter. And most viruses consist of at least those 3 rough components. But then they can have anywhere between 9 or 0 and hundreds of other genes. So there are these massive viruses, particularly in the ocean, that contain hundreds. Herpes viruses, which you might have heard about, that cause herpes or cytomegalovirus or Epstein-Barr virus. They're very huge DNA genomes that encode all kinds of genes, many of which are involved in escaping from the immune response. And for an RNA virus, coronavirus are very large. Most RNA viruses are fairly compact and efficient, but it's 30,000 base pairs, so 3 kb, threefold bigger.
How does that compare with the simple? I think the simplest, the smallest genome is some kind of nematode worm.
I don't know that off the top of my head, but it's much, much smaller. Do you know?
I think 1,000 genes, but I'm not sure.
1,000 genes. Yeah. So, I mean, this would be more like in the tens to hundreds of genes. Mm-hmm. Some of these megaviruses that are only recently discovered, they could well have more like 1,000 genes. I haven't, given that they're mostly marine organisms, it's not my area of expertise, so I don't know.
Now, they can be either RNA-based or DNA-based. Just the ones I know: SARS, MERS, Ebola, flu, hepatitis, West Nile, rabies, polio, and now COVID-19. They're all RNA, right?
Correct.
So is it just my impression? There's a lot more of the RNA-based that hurt us, that are dangerous for us.
I think that's a fair assessment. It's probably about, yeah, there are more classes of RNA viruses that infect humans than the other way around. And I mean, some of it I realized I didn't get to your evolution question, but RNA viruses in general evolve much faster than DNA viruses. And that is because DNA to DNA copying is sort of high fidelity. DNA encodes for the RNA, and it makes, the proteins and the next thing, and it's very easy to copy DNA fairly precisely. We have good proofreading mechanisms. When you go to an RNA virus, it depends on which strand it is, whether you might have to copy it several times or even just once to sort of make more of itself and make proteins. But either way, the RNA to RNA viruses, the polymerases, the proteins that copy RNA, tend to make lots of errors. And so each one of those errors is a chance for evolution. A lot of the variants might never go anywhere because that error might make it non-functional. But some of those errors might be the kind of thing that allow it to skip species. And that might be why we see more RNA viruses, just because, you know, a bat virus should only infect a bat. But maybe if it acquires by random chance a few mutations, suddenly it gains the capacity to replicate in humans.
Mm-hmm. So you would say that RNA-based viruses are more likely to mutate. Is that a fair assessment?
Yeah, they have higher mutation rates, yes.
Yeah, that doesn't mean that the mutation survives, but potentially, statistically.
Statistically, definitely.
Okay, back to evolution. So I'm fascinated by the fact, actually, that was the original reason that I got into viruses. The fact that they're not considered living beings, but they have genes. And you say they don't reproduce. They do. They just hijack a cell, and they do. So in a sense, I see them as a more optimal form of life. They beat us. I mean, we need a body. They don't. They just use bodies. There are millions, billions of bodies out there. They use these bodies. So what came first, the living cell of the virus?
I mean, the ultimate chicken and the egg. I can't say that I know the answer. I mean, the common guess is that, I mean, I would think it's a piece that fell out of a early organism at some point, you know, and could make more of itself. So given that they're dependent on life, I would think that the life had to have come first. Because again, what good would a virus be without its cell?
That's a logical answer. But is it possible that life evolved in two directions? From very simple, it went towards more and more complex organisms like us. And at the same time, it went towards a more and more optimal system, which the most optimal is the virus that doesn't have a body, uses bodies.
It's always possible. I mean, philosophically, it's possible. The one thing that makes me hesitate is the genes that, I mean, the key gene really is the polymerase. That gene that copies the RNA or DNA in order to make more copies. And most viruses have to encode some form of their own polymerase. And those are fairly complex proteins that look like they're hijacked from the polymerases that, you know, required for life at some point and used for their own purpose. So, you know, I guess the real question is, to me, is where did the first polymerase come from? Because without that, there's no replication.
Interesting, very interesting. Okay, let's jump to zoonotic viruses. Now, several viruses jump from animals to humans. I have a long list here: Spanish flu, AIDS, Ebola, SARS, MERS, antivirus, West Nile, Zika, COVID-19. So so first of all, are these the most dangerous? Is this the most dangerous case?
In general, well, I guess it depends on how you do it. For pandemics, most pandemics initiate from an animal to human transmission event. And so yes, animal to human transmission is incredibly dangerous. And part of the problem is that when a virus makes a jump like that, you have a completely naive, unprotected human population. And I mean, this happens all the time, but is often abortive, meaning, so you mentioned Spanish flu, but like avian flu, which was all in the news, what, 5-7 years ago, you know, avian flu is way more lethal than conventional flu or even Spanish flu. However, the avian flus that were jumping directly to people in very close contact with the animals weren't capable of human-to-human transmission very efficiently. There was 1 or 2 potential cases, but it's not been a sustained human-to-human transmission. So there are multiple steps in this process, and that's why most of the jumps occur via mammalian species. It's very rare for something to go straight from birds into humans, because when it does, you see what you get with avian flu is that person might get terribly ill, but at least they don't transmit it to everyone else. But these viruses that jump, yes, they're incredibly dangerous because there's no baseline protection. I mean, conventional flu kills hundreds of thousands of people in the world every single year. But, and we are forgetting about that to a certain extent in the setting of what's happening now with COVID-19. If we had a completely naive population, flu would be just as bad as COVID-19 is now, but COVID we have no immunity to.
Right.
We're protected.
Do we have a sense of how often this happens? How often a virus jumps from an animal to a human being?
I mean, probably every year somewhere it's happening, but a lot of times we don't know what it is. And in fact, there's been a push to increase surveillance of these events in areas where there's close contact between animals and humans, to pick these up in order to assure it. So like Nipah virus, for instance, is incredibly lethal and is another animal to human via weirdly drinking palm sugar and so forth. But
Nipah?
Nipah, yeah. But you'll see cases in Bangladesh and other places. So, I mean, these happen probably, I mean, I don't really have hard numbers. Some of my colleagues would be better people to ask, like Steve Luby, but probably somewhere in the world, some virus is jumping, and most of the time it just ends there and we don't know about it.
Do we have a sense, okay, of course, close contact is one factor, but do we have a sense if there are other factors that facilitate the jump? I mean, we always talk about climate change. Is the season a factor? Are there, I don't know, climatic or other kind of factors that we know could affect? And the second question is, do we have a sense of what triggers the human-to-human transmission? Because, I mean, right? Because if I just get a virus from a cat and I'm the only one who dies, that's it. But then in some cases, I become contagious. So do we have a sense of which factors cause this?
So, I mean, that latter question is the key. And the reality is, I would say we don't have a great sense for what it takes. And in fact, that's been one of a big ethical controversy in science over the last decade or so, as it relates to actually flu studies and, whether it's ethical to try to mutate back and see if you can figure out how to make, for instance, avian flu adapt to human cells. And, somebody did it and then people were worried it could be a mass terror. And should that science even be done? And so forth. So, I mean, we know some things. It has to bind receptors. It has to, be coughed out. But at a very fundamental level, I think that a lot of that is just random chance and mutation, and it just happens to stumble across a solution that allows sustained replication and excretion in humans. And if we understood that better, maybe we would block it better, or maybe we would just have bioterror. I don't know. But there are huge ethical concerns about the implications of doing experiments to do that, which I think has slowed the science of that, which indirectly relates to the conspiracy theories, because
we'll get there.
Okay.
I was just wondering, somebody has done a study where they list the main deadliest zoonotic viruses, and next to it, the place where it started, if we really know where it started, and then, what's special about that place, and see if there are commonalities, if there are,
I mean, usually it's as simple as close contact. And I think that, I mean, for arboviral diseases that are spread by mosquitoes, it's very clear that the climate is having a huge impact, because for instance, the Aedes aegypti mosquitoes that spread a lot of disease are expanding their range dramatically in the setting of climate change. But a lot of it relates to deforestation, which is also intrinsically linked. And as the deforestation occurs, where the animals have much less range, we're encroaching on that much more quickly. I think that you cannot separate the environmental issues, particularly as it relates to deforestation, from the increased rates of zoonotic transmissions we're seeing. I think it's right there.
Very interesting. Okay, now, I'm not sure, maybe you can tell us if the bat is still considered as the most likely animal that gave us COVID-19. But in general, I noticed that several of these come from bats. So, I mean, what's special about the bat? Why it gives us so many viruses, and why the bat doesn't get damaged, doesn't get killed by these viruses? And can we learn something from the immune system of the bat?
Absolutely, all fascinating questions. So bats are super cool, and there are several virologists in the world who for many years have been studying bats and why their immune systems are so cool, and they're in very high demand right at the moment. So first, there is absolutely zero doubt in my mind that this came from bats, because there's a virus that is 96% identical to SARS-CoV-2, which is the virus that causes COVID, in bats. And it's also highly related to SARS-CoV-1, the original SARS, which did it. So definitely bats. Now, was there an intermediate mammalian host? Did the bat infect a pangolin, which infected us? Totally possible. Which the intermediate host has not been identified, if there is one. Usually there is one, but bats are mammals, so it's not impossible that it could be direct mammal. And not to freak everyone out, but there are 500 related other coronavirus species that are circulating in bats just in that region in Wuhan, any of which could make the leap at any time. And again, back to, we haven't the foggiest clue how to engineer one to do that. We just know that it happens, and then life is terrible. So why bats? And it turns out that bats have this weird and awesome immune system that it varies somewhat by species of bat, so it's hard to generalize, but like Ebola was in bat, all kinds of things have been in bat, which is that they simultaneously have an incredibly ramped up innate immune response, this early antiviral immune response that normally we will only upregulate these certain genes if we have an infection. And it's basically genes that shut down your ability to make proteins and other things in order to try and halt the replicative cycle. Bats somehow manage to live with that system kind of ramped up all the time without and yet still manage to function. So that's interesting. But also, once a virus is in them, even though they can suppress it to a certain extent, they also don't develop this inflammatory response that we often develop later that ends up being counterproductive. So, in chronic HIV, it's the inflammation that causes a lot of downstream disease. And bats, once they just kind of live with their viruses, hang out with them, let them replicate. They aren't really bothered by them. So they're both simultaneously very inflammatory and very immunosuppressed in interesting ways that seem to allow these viruses to replicate. And I'm far from a bat immunologist. So though if this keeps up, maybe I'm going to have to learn more about them.
Fascinating. So coming to COVID-19. So the first question, as somebody born and raised in Northern Italy with friends in New York, friends in Britain, Spain, so it's been puzzling right from the beginning that the death rate, now that we have tested enough people, we can't just say, it's it's a statistical accident, that the death rate in Italy was so high, and then in Spain turned out to be as high, in France turned out to be as high, in Britain even higher, then Holland is high, in Germany and Greece is low, very low. In East Asia was low. In New York is very high, in California is low. So the question that many people are asking, starting with me, is it possible that we have actually 2 or 3 different pandemics, that there are different strains and they have different death rate? You rarely see, I don't want to offend Greece, but you rarely see Germany and Greece in the same group. And you rarely see Holland and Italy in the same group. And I don't want to offend Italy. But it doesn't seem to be just about the kind of system, a healthcare system.
I'm not sure I totally agree that it's not about the healthcare system. I mean, so there's a few issues. There is some debate. There's one prevalent strain that was the Italy strain that also hit New York that does have a mutation that may or may not be selected for and more prevalent, that may be slightly better at replicating, and that could be part of this. And that is not the strain that infected the West Coast. And then there's the Asian strain. So there are kind of a few classes of strains. The sort of, we have the most close Asian strain on the West Coast, and then there's sort of the European New York strain. At this point is how it is, and there could be many more. And the truthful answer is we really don't know, because we haven't been able to do the important pathogenesis experiments. Do they really replicate better? Do they do this or that? You know, so far in my lab, we've got just the strain from the first Washington state case. And the reality is everyone in the country is pretty much doing experiments with that strain because it's the first strain we were all able to get our hands on. And we're just now trying to get the Italy and the other strains to do the head-to-head experiments. But it's going to take months. But the commonality between those countries is not just in healthcare delivery, because it's true, some have better healthcare systems than the others. But really, I would argue the commonality is the public health infrastructure and the compliance to sheltering and masking and so forth. I mean, if you look, I mean, Italy just got caught horribly off guard, and it was completely out of control before any testing in an area with a lot of elderly people. And I think that was terrible. And honestly, Spain, sort of the same story. And France, like in all these places, it spread before the alarm bells went off, and to a certain extent in the Bay Area. But, and the New York versus California experience completely relates to hospital overload, right? We have excess deaths in New York because the healthcare systems were completely overloaded, and anyone who tries to tell you otherwise is probably in our federal non-functional government in terms of response. In Asia, you know, you talk to, I mean, again, it's a generalization. You talk to people from most Asian countries, and they're absolutely horrified by the laxity of Americans in wearing masks. And, you know, in Germany, they took very strict measures very early. But I really think the California, New York example is the perfect example of like what public health does for us. We were the first place in the country to shelter in place. We were also the first place with the largest explosion of cases. And we, in the Bay Area, even more so than LA, we have the lowest death rate in the country, I believe. I mean, it's astounding if you look at the Santa Clara numbers. And, you know, at Stanford Hospital, we have not lost a single patient under the age of 75 years of age. Last I checked. I should have checked that again a couple weeks ago. But, you know, as of 2-3 weeks ago. And that is because we were prepared for this huge surge that never came. And so I do think it's a combination of just volumes overwhelming the system, which relates to lack of sufficient public health.
Is it possible, though, that by some kind of coincidence, the Germans, the Greeks, the South Koreans, the Taiwanese have something in their immune system? Like in East Asia, this vaccination is mandatory. What is it? BCG something against tuberculosis.
Yeah.
Is it possible that they have something in their immune system that makes them stronger?
It's possible. It's hard to track it relative to the countries that have done well versus not. I mean, not to be totally political, but if you look, I think so the whole BCG thing is very interesting because it's a vaccine for tuberculosis that people are beginning to appreciate has really long-standing effects, not just on tuberculosis, for which it's a really mediocre at best vaccine, but it really retrains the immune system to be more responsive in interesting ways and protects broadly from death from a lot of things. And there was some brief data which hasn't really followed up. I mean, they're actually doing a randomized trial whether BCG could help, but honestly, I don't think it's the magic bullet. I wish it was. That would be great. It'd be easy. But it's unlikely to be that. Now, is there something else about the immune systems of these people? Absolutely possible. It's hard for me to map any known thing to the regions that have done well versus not. Generally having a female commander-in-chief has been the single best outcome. Germany, New Zealand, you could keep listing.
Germany also has a scientist in
yeah. Yeah, Taiwan, not a woman, but an epidemiologist, happens to be their equivalent of the vice president. Just saying, having science respecting administrations and consensus seems to be the best way to do well in this pandemic.
Yeah, books will be written about the political aspects of this, but let's stick to the science. Actually, let's do the conspiracy theories because we already have 2 questions about this. So, conspiracy theory number 1, it was designed in a lab. How credible is that? And the second one is it escaped from the lab.
Neither are credible, honestly. I mean, it's very clearly mapped out, multiple peer-reviewed papers. Again, the 96% identical virus in the bats. And there's no evidence for the lab escape. That was completely made up.
Okay, good. Do we have a sense, do we have an explanation? Probably we don't have an explanation, but do we have a sense of what could explain the fact that the young people are more or less immune, not completely, for other diseases? The children are the first to get sick in the family, right? And for this one, children don't seem to get sick at all.
That is really one of the great mysteries that will be important to understand. I mean, I would say, get sick is relative. So for instance, with flu and with many diseases, you see a U-shaped curve, meaning that, young babies do poorly, kids get infected but don't get that sick, and then elderly people do very poorly. And we don't seem to see that at all with COVID. There are these really rare inflammatory disorders which are late. And honestly, a few theories have been floated, but we don't know, in part because kids weren't being tested early on. So identifying these asymptomatic kids hasn't really been done, and nobody's studied to see what it is. There've been, some of the theories that have been floated are that there's enough cross-reactivity with the common cold coronaviruses. So there are several species that cause, about, a quarter of the common colds we get. And kids tend to get more colds, and maybe having had a cold recently is protective. So that's a theory. And of course, none of these are mutually exclusive. Another thing that really is interesting is that the receptor that SARS-CoV-2 uses to enter the cell is a protein called ACE2. And ACE2 is actually involved in blood pressure regulation. And we don't really fully understand yet the extent to which having the virus hijack this receptor that's involved in this incredibly complex feedback loop that regulates blood pressure might lead to some of these weird cardiovascular outcomes. But the sum total is kids kind of have a bulletproof ability to manage their blood pressure, right? You don't see kids with high blood pressure. you see older people with high blood pressure. So it may be that they're more resilient to some of these effects on the blood vessels and things like that, just by virtue of being young.
Fascinating. Now, the other question is, some young and healthy people do get sick. One of my friends in Italy spent more than a week in a hospital. And a famous one, relatively young, I think it was 30-something, the doctor, the Chinese doctor, Li Wenliao, I probably mispronounced the name, he died. He was healthy and young. So is that just a viral load, just being exposed to so many cases that caused this death?
Again, we don't really know. That's certainly one theory that's been floated, and it even getting infected, probably it's possible we all ran across somebody at some point in the last several months, and maybe we only got 1 or 2 virions and we were able to clear it and be good. Yeah, the rates of death among healthcare workers were young healthcare. Well, in China, many of the young people who ended up dying were healthcare workers. And so there is a thought that it is a viral load thing because they were the ones facing it the most. And that has been similar in Italy. And that's certainly possible. The other thing is just a lot of the young people who are having complications, it really relates to this hyperinflammation and this coagulation, blood clotting, and so forth. And it may be that these are people who are totally healthy now, but maybe, if they were 70, they would actually have some sort of clotting problem that just hasn't manifested itself, and COVID was enough to push them over the edge. I don't really know, just rampant speculation.
Now, one quick thing about the face mask. I still remember the people on Facebook shouting, "Don't wear a mask, it's useless," and so on. And George Gao, the head of the CDC, the Chinese CDC, in an interview said that the single biggest mistake the West did was to not enforce face coverings. What's your opinion?
I'm with the Chinese CDC on this one. Though maybe not for, they're both partially right. A mask is to me a way of telling your community you care about them. Any sort of face covering is less to protect you than to protect other people from you. So it's not necessarily going to prevent you personally from becoming infected if you're around somebody who is infected. And this is where the misconception lies, right? If I'm wearing a face mask and somebody with COVID coughs on me, well, first of all, unless it's an N95 and really sealed or a PAPR or something, the virus could get in as I breathe. Secondly, how do I take off that mask? Most people grab it, right? Think about what you just grabbed. If somebody just coughed in your face, you've just put it on your hands. So fine if you're going to wash your hands before you touch anything. Less fine if you touch your eye or your cheek or really anything. But probably most people have seen the YouTube videos by now where they show the sprays. What happens when you cough and you're wearing a mask. I mean, they're not perfect if they're not a perfect seal, but wearing a mask massively reduces the amount of stuff you cough out. And that's how the virus is transmitted. So if you cut that by 90%, you can actually think about the math. We know the so-called reproductive rate of this virus is, I don't know what the latest estimates are, somewhere around 2.5, meaning every infected person is infecting 2.5 other people. They're probably encountering many more than that. But if you do a 90% reduction in the air, in how much virus they're spraying, you suddenly drop that reproductive rate to less than 1, which is how you extinguish an epidemic.
Yep. Okay. Well, we Westerners were not so smart after all.
No. Well, and the main reason I didn't wear or recommend wearing masks early on was it really looked like we were going to run out. And I wanted to save them. So that was the other issue, is that we needed to save them for the hospital workers until we could get our supply chain going.
It's
so that was a failure.
Yeah, it's interesting that Taiwan, I think already in January, stopped exports of masks immediately.
Yep.
Okay, now fast forward to June to 2020, and we have hundreds of thousands of infections. Meanwhile, New Zealand just announced they have zero people infected with COVID-19 as of now. And most regions of China apparently are also down to zero. I know South Korea, I think most of South Korea is also down to zero. So the question is there any hope that we, United States and Europe too, will ever go down to zero?
Well, there's always hope. It's a question of how long it will take. So South Korea and China have both had some mild resurgences, which are concerning. The way we get down to zero the fastest is with a vaccine. Of course, vaccines aren't fast. It's at some point we have to balance the risk benefit. We can absolutely reduce transmission through masking, aggressive contact tracing, isolation of cases, and so forth. And yes, we could definitely get down to zero. How well our economy will be functioning at that point is another issue. And that's, I think, where I'm glad I'm not a health economist, because there are also excess deaths from poverty and all the people who've been driven over the edge with this current situation, and that's can't be ignored as much as I might yell, stay at home. Some people staying at home is a death sentence.
Okay, a few questions about testing. First of all, why we, USA, didn't use the test kit that most countries in the world used from the World Health Organization? I believe it was already available in late January.
No idea.
Okay. So there wasn't a very specific scientific reason, right?
Not that I know of.
Wow. Okay, before we get into some technical details, we would like to understand what these tests are. But at this point, we are testing a lot of people. I got tested. I went to San Mateo and I got tested. We should have enough test kits to test people at the airport. If we test passengers at the airport, don't we solve the problem of travel? I get tested before boarding the flight to China or to London. And if I'm negative, I just board and go, just like we check luggage.
It's possible. So to do that, you have to have the rapid test, right? And how rapid, how long did it take you to get your test results?
1 day.
Yeah.
It was pretty I was impressed.
So just, out of fun, so you're gonna chill at the airport for your day? So there are two concerns. I absolutely think, maybe this is an answer, but we have to think about a few things if we mandate sort of testing before activity. And the two things we have to think about are the turnaround time. Well, actually, there are several things. The turnaround time, the accuracy, and the window period. And actually, maybe I'll hit the window period first, because that's the one probably people don't think about as much, which is that period between when you might have gotten exposed and when your test will turn positive. So let's say, for the sake of argument, and I agree, we could massively cut down the number of people flying around with viruses if we screen them. I think the Abbott rapid test has about a 2-hour turnaround, so it's not inconceivable you could, get your test, clear security, get your result before you get it. You would have to get to the airport that much earlier, but it's not impossible to think. There is a national shortage on that particular test right now, but that's okay if we ignore that fact and live in the magical world where we have the test. Or it's the Cepheid test, whatever. I can't keep them all straight. But what if you had been to a party the night before and been exposed? We don't know how long actually it takes for you to turn positive, but probably on the order of 4 days-ish. So let's say you're asymptomatic, you test negative, you're quietly brewing the virus at some low level, and then you get to your destination.
Yep.
So that wouldn't happen often, but it could happen. And hopefully we'll have more data. I mean, this is also new. There's so much more that's unknown than known. I don't know what that window period is. But if we knew what that was, we could better make an assessment of what that risk is.
So, but if the window is days, then we will never have something like the home pregnancy test, where you can just test at home and you know yes or no.
Well, I mean, we absolutely could have a home pregnancy test, yes or no. It's just you have to always know, just like a pregnancy test, you don't take the pregnancy test the day after. You take it after the baby has grown big enough that there's the hormone that can be detected on the pregnancy test, which I think is usually like a week. I can't remember. So there's that window. It's been a long time since I thought about pregnancy tests. So there's that window period in there that is what takes into account. So, you know, any test, you have to know that that test, so actually for the antibodies, we kind of have a better sense for that window. It takes a minimum of 2 weeks to develop antibodies. So any given antibody test that you receive doesn't really tell you much about how you're doing now. It's telling you what you were 2 weeks ago, which is a pretty long window.
Yeah.
And that's at the short end.
Yeah. Okay, can you walk us through the existing tests, the molecular test, the PCR-based test, and the serological test? What's the difference, what we get from one and the other?
Absolutely. So the standard test that probably many people have had, the nasal swab that feels like it's going into your brain and is quite painful, is testing for that nucleic acid, that genome of the virus, which is RNA. And it's, in general, it's funny, there's a lot of sensitivity and specificity numbers that are thrown around. So sensitivity meaning the likelihood that you detect somebody who's positive who really is positive. And the reality is, to know that, you have to have a gold standard. And it's hard to know what the gold standard is. So right now, the gold standard is repeating that test enough times and hoping and getting a sense for it. So we think they're reasonably sensitive. And in general, nucleic acid tests like this, because they can find a very small amount of RNA and make a bunch of copies of it, are very sensitive. So most of them have a sensitivity that's quoted in the high 90s, like 97%. In addition, the specificity is very, very good of these tests. And the specificity meaning how likely is it to pick up something unrelated and tell you it's COVID. So for instance, a common cold. It's the same class of viruses. Like, what are the chances that you just have a cold and they detect SARS-CoV-2 and tell you have COVID? And actually, they've very carefully designed the way these are amplified that really hasn't been an issue. But I would argue that we don't truly know the sensitivity numbers because we don't have a gold standard per se, and we only know what we can detect. We do know it's more sensitive than, say, trying to culture live virus from the nose, because a few people have done that where they've taken swabs every day and people were known to be infected and seen how long you detected it. And you can detect the RNA long after you can actually culture live virus out. And we don't know if people are infectious during that period, whether that RNA is just a reflection of old non-functional viral particles, which I think a certain amount might be, or old virus, and those people are probably no longer infectious. But out of an abundance of caution, we consider anyone who tests positive. So those are good tests. They, again, you have that window period. That it's possible that you just might not have enough virus that people can detect it, or that the virus goes away and you had it. So that's the SARS-CoV-2, the diagnostic test. Then there's the antibody tests, the serology tests. And those are detecting, instead of the virus, your immune response to the virus. So specifically the antibodies that bind to the virus. And interestingly, with this virus, those antibody responses tend to be pretty delayed. So really not seeing significant antibodies until at least 14, 21 days post-infection. But the nice thing is it tells you that somebody's been exposed to the virus. So anyone who has a positive serology has been exposed at some point in the past. And again, sensitivity, hard to say. Not everyone who's been diagnosed with COVID has been developing really great antibody responses, and we don't really understand why, but most people do. Very good. And specificity, again, they've designed these to try to not cross-react with the common cold viruses. So they're all pretty good tests. Now, in the early phase of the serology tests, there were a bunch of not very well validated test kits out of China that weren't very good and had horrible sensitivity and specificity.
So how accurate are the antibody tests we have today? When New York just did a massive study and came up with an estimate that 10 to 20% of the population has been infected. How good is that result?
It's very good. So that was done with one of the high quality serology assays. And I think there's sort of old school ways to do it by an ELISA, and then there's these lateral flow, the pregnancy test like things. Which are a little less sensitive, but are still pretty good. But the New York data is pretty solid.
Now, can we use people's antibodies to make a treatment?
It's being actively explored, and it's quite possible. There are tons of people who've been infected. So there are two approaches to this. First, one is kind of the crude approach, which is you just take somebody who's been infected and you grab their blood and you purify the plasma, which is full of these antibodies, and you infuse that in somebody who has COVID and is doing poorly, and that's convalescent serotreatment. And there's an ongoing study of that. It's not a randomized study, I don't think, here at Stanford and at many other places. And so far it appears to be safe and maybe beneficial, but it's hard because I haven't really done the randomized controlled trial to see if it really is just that we gave it to so-and-so and not to so-and-so. And the other approach is to, which is a little more targeted, is to actually clone individual antibodies out of people that have really strong activity against the virus, and then make those antibodies in the lab very pure one antibody. So it's not this messy sera with everything in it. It's just the one antibody. And that is going into randomized control trials. Tons of companies throughout the country, as well as academic labs, have been cloning these kinds of antibodies. And several are in trial. We don't have any results back yet. But
when you say cloning, it means they're making synthetic ones?
Yes. Yeah, so what happens is you find the B cells that encode the antibody. People isolate those, find the genes that encode for the little antibody molecule, express that in another cell, make it into little antibody factories, purify the antibodies, and give them to people. Some of the Ebola treatments were done that way too.
Now, it's treatments. Quickly about treatments. Every time I read a report that says this treatment looks promising, a few days later I read a report that says, well, no, unfortunately, no. What's the situation?
It's kind of a mess. The situation is that this is a really new pandemic, and we don't have a lot of rigorous randomized control trial data, which is ultimately the best way to assess something, is to randomly assign and make sure that the outcomes are good. So one exception is remdesivir, the Gilead drug that everyone's heard about, where it was a nicely done randomized trial as well as several observational trials, actually. And it showed that people got out of the hospital faster if they were randomized to remdesivir. It's not a perfect drug. It's got some toxicity. It has to be given IV, but it is now the standard of care for severe cases is to give remdesivir. The whole hydroxychloroquine and azithromycin mess is pretty much a mess. And I don't think we know because, unfortunately, it does inhibit the virus in a tissue culture dish, but all kinds of things inhibit viruses in tissue culture dishes. That doesn't mean they're going to work for people. There was the initial, I think it was the French study that showed a benefit that was fatally flawed. They conveniently left out the people who died on their hydroxychloroquine arm in their analysis because they didn't stay in the regular hospital or they went to the ICU. They removed them from there. I mean, it's entirely inappropriate and fraudulent, and the study, if it hasn't been, has been retracted. So the study that supported the benefit was retracted, but there's still reason to think it could be beneficial. There were a bunch of randomized controlled trials going on, and then those two big studies coming out saying it was not of benefit, which have since been retracted. So now the randomized controlled trials were stopped, and we don't know the answer. So I think people are trying to get the trials going to see if there's any benefit for hydroxychloroquine again. There were several studies, and for many of these things, it's important to note that the drugs that work at late stage disease might be different from those that work at early stage disease. Like hydroxychloroquine might actually prevent acquisition, but might not be good for treatment because it has bad cardiac side effects. But if it keeps you from getting the virus in the first place, maybe that's not so bad. And that's what the trials that were ongoing were investigating, mostly healthcare workers and highly exposed people, randomizing them.
Okay, so we have a question, actually, it's a good question. Will antibody treatment be available before a vaccine? I mean, could that happen, that we have a treatment that is that makes the vaccine less relevant?
Not practically. This is an idea that's been explored in the HIV field because, for 40 years, we've been unable to make a vaccine for HIV, but we do have a few antibodies that we know can neutralize the virus. And there've been a few trials to just make people make those antibodies through gene therapy. But it's not a very practical or scalable solution because it is gene therapy. To do that, the approach used in HIV was to inject virus that encoded for the antibody into people's muscle where it will live for 30 years and secrete antibody. And at least those trials didn't have a so-called kill switch. Like, what if that antibody starts to cause an autoimmune disease? I think you kind of need a way to shut it off. And it's also absurdly costly to do gene therapy. So it's not a worldwide scalable issue. We really need a vaccine. And, frankly, the Chinese studies of the just inactivated virus that they injected looked promising, and that's far more scalable. Grow up a whole bunch of virus, inactivated and injected in people.
Okay, let's get to vaccines. This is probably going to be the longest section of our conversation. So more than 100 vaccines under development, vaccine candidates under development. And unfortunately, you have to give us some education on vaccines in general. We hear of attenuated vaccines, inactivated, you just mentioned, recombinant, mRNA, DNA vaccines. Can you walk us through these families of vaccines? Tell us which ones are real, which ones we have used already, and which ones are just brand new, and pros and cons and so on. All yours.
Okay, that's a loaded. So vaccinology is an interesting field in and of itself, and honestly, historically, has been more trial and error than anything else. So the simplest is the one I alluded to already, which is an inactivated vaccine, which is literally where you take the virus and you treat it in some way to kill it. So I know I already told you viruses aren't alive, but but you kill its ability to replicate itself. So typically this is done with things like just formalin fixation that screws up the proteins, or things like this that, so it's fairly crude. Sometimes the ways you fix it can make it not look like the real virus, because of course you can't have it look exactly like the real virus, or it might be the real virus. But those are the simplest vaccines to understand, and you just inject that into people.
And we've done this many times.
This has been done, yeah. Most vaccines we use today are not done that way because they're really crude, tend to have a lot of off-target effects, and so forth. So most have been replaced by live attenuated vaccines, and honestly, the first vaccine was a live attenuated vaccine, if you think about it. So that is exactly what it sounds like. Live meaning it has the ability to replicate, but attenuated meaning it kind of stinks at it. So Edward Jenner famously noted that milkmaids didn't get smallpox, but mostly it was the milkmaids who had the scars because they'd had cowpox. So cowpox, otherwise known as vaccinia virus, is highly related to vaccinia, the virus that caused smallpox. But it doesn't really replicate very well in people. It causes these scars, these local issues, but it doesn't get into our bloodstream and make you sick and die like smallpox does. So he famously gave, after experimenting with a few people, like and this was done in China too in ancient years, they would blow the scabs onto people and let them inhale it. Actually, long before Jenner. But the difference, what Jenner did was he not only challenged a kid with cowpox, he then intentionally exposed that kid to smallpox and injected him with smallpox, and the kid survived. And that's why vaccines are called vaccines, for vaccinia, which is the name of cowpox. Mm-hmm. So live attenuated vaccines have been around for as long as we've called them vaccines. And they can be quite effective. So the oral polio vaccine is a live attenuated vaccine. And there are many ways that you can think about inactivating them. You passage them through different animals, you do all kinds of rigmarole. The danger of live attenuated vaccines is occasionally they can revert to become a problem in and of themselves. And this is what has happened in a small number of cases of polio, where it can acquire certain mutations in replicating in people and then actually cause local small outbreaks. So live attenuated vaccines are by far the most effective vaccine in preventing the infection, because you get this really robust immune response because you have it replicate and do all that. But the safety profile isn't quite as good, because there's always that risk at the back of your mind. The most promising HIV vaccine candidates that were tried in macaques were also live attenuated. They were great, except that there was reversion at a certain rate in animals developing AIDS, and we don't want that. So they can be great, but probably the slowest to get to large-scale trials in some regards, because you need to watch for a long time to see that safety signal.
Right.
Would you feel safe getting a live attenuated vaccine this week? Not so much.
The word live is kind of scary.
Yeah. So in modern times, we've moved a lot to these recombinant vaccines, where instead of just taking the virus goop and killing it, or live attenuated, you express certain proteins from the virus, particularly those envelopes, which are how they get in, and you use that as the vaccine. And that's incredibly safe because it's just the protein usually hitched onto some complex that holds it. Incredibly safe. A little more expensive to make. So for instance, hepatitis B vaccine is one of these. It's a conjugate vaccine. There are polysaccharides and so forth. And you can even get fancy and make virus-like particles out of them, make little empty virus capsid-like things, because they'll self-assemble with the proteins you want. And some of the HPV vaccines are made that way. And they're really great because they look like a virus, but they are entirely safe because there's no nucleic acid in them to cause replication. And then you get into these newer platforms, DNA vaccines, where you just inject DNA and with the hopes that the DNA gets into a cell, transcribes into RNA, makes a protein, and then that cell itself is making the viral proteins and stimulating the immune response. And those have been less tested in human, but they seem to be pretty good. They have the advantage DNA is quite stable. It can be made very quickly. You want to talk scalability, how to get vaccines to the world. Great options to do a nucleic acid vaccine if you could get them to work. They so far have been kind of meh in people, as near as we know, but Dan Baruch and his colleagues in Boston published a DNA vaccine for COVID in macaques, and the data look very, very promising. And then the final is the RNA vaccines, of which there are a gazillion in trial already. And everyone heard about Moderna and their New York Times headline for the 8 people they injected with their vaccine, so a little hype there. Works with a similar principle to DNA vaccines, except it's never been tested in people before now. We have no known vaccine candidates that do it. Very promising potential approach. The biggest issue is the stability of RNA, which is horrible, which is what Riju Das is working on and hopefully will solve for us, because the little secret about the Moderna vaccine that I've heard is that it had to be stored at -80 Celsius until about 30 minutes before being injected. So that's not a terribly scalable option. So hopefully Riju will figure out how to stabilize these structures so they don't require refrigeration, or at least only require refrigeration.
So DNA vaccines have been used on humans. We have some.
At least in trial. I'm trying to remember if I should know this, if a currently licensed DNA vaccine exists, but they've certainly been in trials on people.
When they talk about adenovirus vectors, is that for recombinant vaccines?
So those can come in 2 classes. Those are mostly a recombinant attenuated. So the adenovirus is one of the common cold viruses aside from coronaviruses. And they have a fairly big genome and they're pretty easy to manipulate. So people will cut out the adenovirus parts and put in the gene of interest and put that in people. And those are being used in combination with DNA vaccines. For instance, Dan Baruch's data at Boston often is an adenovirus and then a DNA in conjunction. Like you give one and then you give the other, you give them both at the same time. And those can be quite effective as well. But they're usually messed up in some way. Some of them can replicate, but most of the ones that have gone into people, they've made so they can't even replicate. So it's an actual virus particle, but the genome is missing a piece that allows it to make more of itself. So they're not truly even attenuated because they can't replicate at all, most of them.
Okay, now of all these categories, if I understand correctly, we have at least 3 problems. 1, how long it takes to develop it. 2, safety, how safe is it? And 3, scalability. I mean, once we have a vaccine, can we make billions of it? So if you look at these categories, when you look at these categories, where do your guts take you? Which ones are more promising today? And I know this changes every week based on what gets discovered and published.
Yeah, I think it does change every week. I mean, I think the real answer is that we probably need to pursue multiple at once, in part because I do worry about the scalability a lot. I mean, I'm an ID doc. I've spent most of my life studying diseases that don't affect the US, that affect, other countries that are less served. And, I think, maybe, if we can get an RNA vaccine going here, that's great. Sign me up. I'll take it. But I'm going to need to see the DNA or the inactivated vaccine for my colleagues in South Africa or Kenya. So I honestly think we need to go all in on all of them. and probably set up manufacturing centers in different places and tune the type of vaccine to the cold chain issues in a region.
Yeah, we heard also Bill Gates and others talk about the issue of scalability. But I'm also worried a little bit that the political pressure to come up with a vaccine could give us something that is not safe. I mean, if something is announced in record time, I don't know if I will trust it.
Well, I think the whole hydroxychloroquine debacle tells you what you need to know about that, right? I mean, everyone else, we all have whiplash. That is a concern. You know, me personally, I think live attenuated is going to be, again, I stated pretty clearly, the most effective at getting a great immune response. But I think that will be the slowest to know what the safety profile is. I feel pretty comfortable with the safety profile of a recombinant or a DNA or even an RNA vaccine, in that we have a lot of experience making these. They're pretty clean preps generally, and it is just, you know, one shot that's going to target immune responses. Now, there's a trade-off. The more focused you get in your vaccine target, you know, if you just focus in on the spike protein, the safer the vaccine is, but potentially the less efficacious it is. And this is what's going to be interesting, because we don't know yet what protective immunity to SARS-CoV-2 looks like. We do know that people infected with MERS and SARS-CoV-1 developed pretty good antibody responses that appeared pretty long-lasting, and we think were protective. It's been very difficult. Like, we don't have a vaccine for the common cold. And it's partly because it's very difficult to get a vaccine that will get good immunity in the upper respiratory tract. And even this data I'm talking about, granted it was monkeys, but not people, but it's a pretty good model. The vaccine was much more effective at limiting viral replication in the lungs than in the nose. And this is going to be a thing that we're going to have to deal with. It may be that we will have a vaccine that prevents disease, but not transmission. Because most people get sick when it gets lower, but most people transmit because it's in our upper respiratory tract. And my hope is that won't be the case, that we'll be able to sterilize the nose, but you know, we don't yet have any vaccines that actually induce what we refer to as sterilizing immunity, meaning the virus just can't get in the nose. But we also haven't tried very hard because they're not like high priorities. So don't know if it's impossible, just we don't know.
So we had a couple of questions about, which tells me that there is some anxiety out there about this, about who will get there first. We didn't mention which groups are working on which kind of virus. Would you agree that the Chinese tend to work on more proven technology, and here in the US we are trying more innovative technology? And what about Europe? I heard of projects in England and Germany. What's your feeling from the point of view? Which groups you follow more?
You know, again, I sort of feel like we're going to need a multi-pronged approach, because it may be that we're going to go through phases of this, right? If the initial vaccines only protect from lower respiratory, maybe with a newer approach we can, you know, stop transmission in the long run. But so I'm okay with a stepwise approach. You know, I am a little concerned that the US isn't prioritizing, you know, some more simple approaches, but again, I mean, we also generated the Hpv vaccine, which has prevented, you know, probably millions of cervical cancer cases. And that was, you know, a high-tech approach to something that nobody had been able to get mucosal immunity, good immunity in the lower genital tract for many years. And it took, you know, a long time and a lot of development. So I don't think technology is bad here. I think it's complementary, and we'll see which ones lead to the best response. The problem with the crude approach is that you can end up with all these off-target things that it's not as protective, because you do denature the proteins when you make those. Right. And sometimes that doesn't lead to a good immune response.
I guess we forgot the fundamental question. How long does it take normally? How long, I mean, what's a normal time to quote unquote market, to mass availability of a vaccine?
So I think historically the fastest ever to this point has been 5 years. There have been many advances in vaccinology that will allow us to go faster, but the fact that within a week of the sequence being published, Moderna and other companies were already making a vaccine. We moved at warp speed, and these new, we'll see how well they work. These new DNA and RNA platforms have the advantage that we don't have to do all the trial and error of like, does this protein work? Does this protein work? Does this, you shove it in and it's incredibly fast. So we saved a ton of R&D time. But what is still going to take time is you gotta go through the animal, the phase 1, the phase 2 safety, and that I don't see that we can do that in under 12 to 18 months, even in the best case scenario.
Now, there's a case where we haven't succeeded in 40 years, right? 8, we still don't have a vaccine. Why is that? 40 years.
Yeah, believe me, and I've spent 20 of them working on that. HIV is a very different beast. So we mentioned RNA viruses mutate, and coronavirus is actually much less than most RNA viruses. They have a proofreading mechanism. They still mutate. HIV mutates massively. But importantly, it has a remarkable ability to evade the immune response naturally, the way it attacks, and it's just been really tricky. No one has ever naturally, that we know of, cleared HIV infection. Most of the virus, measles, you'd rather get the measles vaccine than get measles, but most people who get measles do recover. People don't recover from HIV, so it's been hard. But the biggest issue is most effective vaccines rely on so-called neutralizing antibodies, the antibodies that bind to the surface of the virus and prevent it from entering the cell. And it turns out that HIV is just a master of disguise from neutralizing antibodies. Unbelievably good at it. So the early vaccines that went in, they could make neutralizing antibodies to that one strain, but they're like a trillion other strains out there that could just ignore those antibodies and get through.
So you see
I'm less worried about that for COVID.
I'm sorry?
I'm not as worried about that for COVID.
Right, I was going to ask you, does this coronavirus look as tricky or more manageable?
It does not look as tricky. I'm quite optimistic.
Okay. Now, not political questions, we don't want to get there, but the United States is reopening, and at a strange time, in a sense. I mean, Italy and other countries in Europe and in Asia have reopened when the number of deaths had declined dramatically. I mean, Italy now has, 50, 60 deaths per day. United States is still, 1,000? So you think it is the right time to start reopening, or are we, again, making the mistake of taking it too lightly?
I mean, probably taking it too lightly, although I think that arguably should be a regional decision too. There are places in the country where the cases are going up very high, and there are places like, I mean, if we took New York out of the model of the US cases, the US looks horrible. But cases are slowing in New York. So I'm quite nervous about reopening. I do think we're going to see a second surge. But I also, really do believe that masks work. And if people wear them, like even all the time, which nobody likes to do because they're really, like, I hate wearing a mask as much as anyone else. I think if we could do it in an intelligent way and be very cautious about how we do it, then hopefully we can do some reopening. But I am quite concerned from a public health standpoint that we don't have this under control yet, and we don't have the testing capacity, and that we could see a second wave.
Okay. Now, let me also summarize a couple of questions, since I'm sure at some point we have to end this. You have other things to do. So in my opinion, one problem in the West is that we assumed best case scenario. We assumed the virus will stay in China, will never come here. If it comes here, it will not be a big deal. And let's face it, it's not a matter of right or left. We always sort of over-optimistic about the situation. Now, can we plan for the worst case scenario? Worst case scenario, the virus doesn't go away, keeps killing people, a second wave comes, and we don't get a vaccine for, I don't know, 2, 3, 4 years. If you had the power, what kind of investment, infrastructural investment would you do to face the worst case scenario that, unfortunately, I don't hear anybody talking about? But that's not a good way to be prepared for the worst case scenario.
Well, I mean, that's a big question that could take a full hour or two on its own. I mean, we need better masks. We need better hospital capacity and ventilators. We need more personal protective equipment for healthcare workers. Ideally, no one in New York wearing trash bags. So, I mean, those are sort of structural things. We need to have the hospital capacity to deal with this. And, I mean, we prepared for a huge surge here that we never really needed. But I think that's why our local rate was low. So that's some of it. But the other thing is, the luxury that we've had with the slower rates of infection around here are that we can actually do some of the science that might help us develop new treatments and so forth. So, if I had my magic wand, nobody wants to do this, but the convalescent sera question, like, that should be a randomized controlled trial, not a like, oh man, this person's sick, let's give it to them, because what if we're making them worse? We need to know that. We should increase our infrastructure in the BSL-3 style labs that allow us to do these drug screens. I mean, I get 20, 40 people who want me to test drug A or drug B, which they think might target the replicative cycle and so forth. But like, there's only so much lab capacity we actually have to do these drug screens. So that would help. But I mean, I think the biggest thing is that we need a good public health response. I think hopefully, if we've learned nothing from this, it's that, years and years of ignoring public health is been to our detriment. And, not all of us ignored it when we heard the reports out of China. I had my fellow go down for BSL-3 training at the center in Irvine that does that, right? Ostensibly to work with something else, but it was always in the back of our mind that might not be what he did. And in fact, he's got a SARS.
Is anybody investing in better masks, face masks, since that seems to be something relatively trivial that could lower the number of infections? So why not invest in making masks that are easier for people like me, for idiots like me, easier to wear and to manage and protect more?
I think a lot of people are working on that in various aspects. I mean, I think I had to say in the first weeks after the shutdown, I think every engineer at Stanford contacted me also about wanting to work on decontaminating the masks safely and so forth. So, I mean, I think that would be great. At the end of the day, any mask that is actually effective at blocking transmission is going to be uncomfortable. because it's by definition limiting your airflow, which doesn't feel great. But yeah, I mean, there's a million more things I could say, but I think that would be a part of it. You know, and I also want to mention that we really need to involve, I think the hardest hit group in terms of the response to the pandemic have been I mean, it's been super hard on the doctors, but like the nurses, the respiratory therapists, everyone else, I mean, it's just been very stressful. And if you know, look at what happened in New York.
Absolutely.
It would be great if we have that surge capacity to bring in people and properly protect them while they're at work.
Absolutely. Okay, I think we used enough of your time. Thank you very much. This has been incredibly educational and informative. Thank you for your work, for your research. Thank you for your time. Everybody else, please wear a mask. Thank you. And so for the audience, yes, I'm going to post the recording. Hopefully, I will not screw up now that I end the meeting, and I will post it probably on YouTube. In 2 days, we'll have Mark Jacobson talk about renewable energy, and then in 5 days, we'll have probably a very interesting topic, robots and COVID-19, where we'll see how robots can help in this pandemic and you know, future ones. Okay, thank you, Catherine, and thank you, everybody. Good evening. Bye-bye.